Hello and welcome to the Future of Health presented by Abbott. You are on TEDx, India's largest ideas platform, and I'm Govindraj, Ethiraj. The Future of Health focuses on aspects of well-being, medical discoveries, and technological advancements that are improving the quality of life. All very important, you will agree, at a time like this when many of us are still at home in about month 5 of a lockdown of various sorts across various parts of the country in India. So now let's talk about technology, which is fundamentally increasing and driving the new innovation that we are seeing, particularly in healthcare. Think about this. Once upon a time, not very long ago, a doctor would take maybe hours to go through a handful of X-rays. Today, thanks to artificial intelligence, a computer can scan hundreds of X-rays and find out aberrations and do all of that in maybe even minutes. So diagnosis is faster, and so hopefully is the cure. Now, this was a very simple example, which I hope you will understand that like I did. My guests today, however, work on something far more sophisticated on the cutting edge of health technology. Whether it's using artificial intelligence to mine diabetes data or developing organ-on-a-chip technology, integrating stem cell research and tissue engineering. On that note, I am pleased to welcome our first speaker for today, Dr. Ruchi Sharma, CEO and founder of Innovators. Ruchi obtained her PhD from the University of Edinburgh, working on stem cell research, and subsequently held postdoctoral positions at the Rossendale Institute, University of Edinburgh, and the Wellcome Trust MRC Cambridge Stem Cell Institute. Ruchi was nominated by BioBeat as one of the 50 leading female scientists and innovators in healthcare in 2017. Following her time in academia, she started her own biotech company, Innovators, which focuses on development of organ-on-a-chip technologies, integrating stem cell research and tissue engineering. And I'm sure you will wait to find out what this means and how this impacts you, like I am. In developing these new technologies, Innovators aims to provide a patient-tailored drug discovery and screening tool that is more physiologically relevant, cheaper, and ethical compared to animal models. So on that note, let me first hand over to Dr. Ruchi Sharma, and it's over to you.
Thank you, Govind. Good afternoon, good evening, and good morning, wherever you are. Next 15 to 20 minutes, we are talking about the future of healthcare. So I'll be just sharing my screen with you. Human need and curiosity are drivers of innovation. Automation, robotics, photonics, electronics, biotech. These are shaping our healthcare. The future starts today, not tomorrow. The year 2020 brought a fair share of challenges for the world. It's heartbreaking to see the loss of precious lives and livelihoods. We live in a connected global community, and together we are seeking ways to keep our loved ones safe. No doubt, Google Trends show a newfound interest in coronavirus diagnostics, treatment, and data to know what's safe and what's not. Innovators is on a mission to provide innovative technologies for drug discovery and diagnostics that is precise, but also available and affordable. Data defines our decisions. Decisions define our actions, and actions can change the world. But the way we see data makes a difference. Innovation, as is happening for COVID-19, will change the diagnostics for several diseases. The most reliable method at present for testing is polymerase chain reaction. And we owe it to Hargobind Khurana and Kary Mullis, both Nobel laureates. They figured it out that a genetic code works like a zipper. with the teeth, ATGC. He made the zipper head for fast alignment. And Kerry found out that by making it hot and cold, we can zip and unzip these faster and increase the numbers enough so that we could detect. So this schematics is actually showing the essence of life. The coding and decoding from our genetic information into proteins and our functionality thereof. So what's the problem with the technique which has evolved and has led to so much of development in our understanding of our systems? So the problems is the mass testing, the requirement of skilled personnel, the multi-step procedures, the small volumes, and infectious diseases. So what's happening? STEMNOVATE developed a rapid testing technology, bringing together bioinformatics, molecular biology, sequencing, computer-aided designing, simulation, and optics to deliver a simple solution that could be employed in fast-paced environments like airports or far-off places like remote laboratories or clinics. Instead of primers, we created probes, which are specific to the virus nucleic acid. And these can bind with huge complementarity, so there is alignment. And because this reaction is isothermal, it does not require this hot and cold cycles. And as the readout is optical, it is a rapid test. And as the reaction happens on a chip, it allows library preparation itself over in that device. And this can then be taken further for sequencing, and you'll find out what virus it is, how it is mutating, what kind of infection it is creating, a perfect way of finding out, tracing and tracking the infection. So in drug diagnostics, what's changing? So the future will be of rapid, scalable, affordable methods. It's a change and it's happening now. COVID-19 is also disrupting our drug discovery methods. We are now repurposing, innovating to do in months what it takes 15 years and billions. So in the AMBER preclinical phase, predominantly, we try to find out how a drug will work. That's the mechanism of action. And it is usually studied in cell lines. The promising candidates from this stage progress towards animal testing, and that is to find out how the drug will be absorbed, metabolized, distributed, and excreted. In the clinical phase, thousands of normal patients, and then the patients are tested to find out and profile the safety. Unfortunately, that's where most of the drugs fail. So let's look deep into what's changing with the focus just on drug metabolism. So we narrow it down to one organ where it all happens in our body, that's liver. As most drugs get metabolized here, the enzymes which are involved in biotransformation, we talk about them, they are the cytochrome P450s, and the relevance to drug metabolism and the pharmacogenetics. So usually, we will study 2 or 3 of these enzymes. We look at the proliferation profiles. We look at inhibition or stimulation of these cytochromes. But what they miss is the extensive interplay of interactions among several enzymes in this family. If we ignore all diversity and look just at the gender differences, females have 2-fold higher expression of relevant cytochromes. And this contributes to higher toxicity and adverse drug reactions in females. So the following represents the real liver whole transcriptome. So all the red blobs that you see, they represent genes which are involved in drug metabolism in a particular moment. So let's curate. So this is an example where there's a higher expression of one of these cytochromes, CYP2E1. So it metabolizes ethanol, alcohol, and it also contributes to metabolic diseases and diabetes that Deborah will be talking after my talk. Another cluster, if we open up, we see there is higher expression of another enzyme, ADH. Now, this is also involved in alcohol metabolism. Now, this individual has higher chance of toxicity at a concentration of drug that we can normally all tolerate. So inferring this information without knowing even the clinical history is highly significant. Now think about the cancer. We can find about new targets, new biomarkers, and the treatments. So our limitation over here is the availability of the liver to study such interactions early on in drug discovery process. So here I'm introducing the regenerative medicine field, the field of stem cells, and a very exciting and very, very innovative technology of cellular reprogramming. Professor Yamanaka and Professor Gurdon were awarded the Nobel Prize in Physiology for this in 2012. So what's unique about this technology? So at the early stages of our embryonic development, the cells can renew. They can form the same cells with the same kind of characteristics again and again and again. And then these differentiate into multiple cell types. They can form neurons, they can form heart cells. But as these cells differentiate, they lose this capacity. But there are a lot of ethical questions around embryonic stem cell research. So what's unique about cellular reprogramming is that we induce the genes which are expressed in embryonic stages and make the adult cells behave like embryonic stem cells. So these don't have the ethical issues as of embryonic stem cells because no embryos are destroyed in creating these cells. And we characterize these cells. So they show the same kind of expression as is there in the embryonic stem cells. And because of this, we can now guide them to become different cell types. So here we have shown the guided or the directed differentiation of iPS cells into liver. So over a period of time, several studies of the embryonic cells and the fetal development, we know the kind of gene expression, growth factors, which lead to this state-specific development. My co-founder, David, in Edinburgh, has an active group that's been working for last 20 years in this field. So the methods that we have, they are now for bioproduction of these liver cells. And we are simulating the liver development. So we evaluate cells for expression of key markers, protein expression. We study the matrix. These are the proteins which the cells uniquely secrete. And understand the tissue organization, the multicellular functionality, including interaction with immune cells. So the microengineering, microfluidics, electronics, that is to achieve the automation and faster data analysis with the integrated pharmacogenetics. So what is the significance? So the importance of drug metabolizing enzymes is important to determine the safety of drug, but also to reduce the adverse drug reactions. And this is represented in the expression of certain cytochromes that I just talked about. So if we take example of some of these key cytochromes in Caucasians, the population, about 9% of the population shows low expression, but in African population, about 22% of the population has lower expression of these key metabolizing enzymes. And interestingly, the Asian population has 50% lower expression of these enzymes. That's why we see the huge variability in our drug response and also the toxicity. Similar trends are becoming apparent for coronavirus infection and the response as well. So I'm just showing you a video in that you can see what the technology, so we use sequencing, transcriptomics, the real-time analysis. So here you can see these are the skin cells. So as these are proliferating, you can see them growing, increasing in numbers. So these are proliferative cells that we induce the cells and they become induced pluripotent stem cells. Now they have the characteristics like embryonic stem cells. These are directed, differentiated into liver cells. So this unique epithelial morphology, hexagonal cells, they are the characteristics of the liver cells. These are the cardiomyocytes. They are expressing the unique markers that are expressed in the heart cells. And you can see them beating. That's to determine their functionality. And what we do with these cells, it's for computational modeling. It's to analyze their behavior, the changes, and to analyze them faster and better. So the future of drug discovery, what it looks like, it's about automation. It's about faster and cheaper sequencing. It's about computing. It's about developing these integrated circuits and models to reduce the indirect techniques that we've been using, reducing the use of animals. So I'll emphasize here that the change that is happening, it's for better. It's for better healthcare. It's for future medicines that are going to be more affordable and safer. And I'll end the talk with Bruce Springsteen's quote, "Nobody wins unless everybody wins." Thanks to Team Innovate and our funders, Innovate UK and Angels. And I'll be very happy to take any questions after this talk. Thank you.
Thank you very much, Dr. Sharma. So it's now over to Dr. Deborah Wake. Let me introduce her. She's the co-founder and CEO of My Way Digital Health. She's also a senior clinical lecturer at the University of Dundee and an NHS diabetes consultant. She is a national clinical lead for internationally award-winning and cost-saving The My Diabetes Way, which offers online data access and education to all patients in Scotland with diabetes. And the prospects for that are immense, particularly if you were to extrapolate that to countries like India, which have a very, very large diabetes base. She founded My Way Digital Health in 2017, co-founded to commercialize My Diabetes Way with Scotland. She's won several business entrepreneurship awards, including the Scottish Edge Bio Dundee Venture, and was a top 3 finalist in Accelerate Her and Women Ahead. Dr. Wake graduated from the University of Edinburgh and also gained a PhD in Diploma in Medical Education. It's over to you, Dr. Wake.
Many thanks for the introduction. So I'm going to be talking about the future of healthcare, but with a slightly different slant, talking about the potential for data and digital innovation to drive healthcare improvements. And I'm going to focus on non-communicable diseases or long-term conditions as an exemplar, but with a particular focus around diabetes, which has been the area that I've worked in for many years. So diabetes, I would suggest to you, is probably one of the biggest health threats facing the world. And that's quite a big statement to make when we're in the middle of an infectious disease pandemic. But diabetes has been silently killing people for many, many years with vast numbers of completely preventable deaths and morbidity. Yet there's been no global emergency or call to action that we've seen with COVID. But through the data I show you, perhaps there should be, and perhaps we should be accelerating and innovating more in how we are delivering care for this condition. So around 10% of the world now has diabetes, and the rates are even higher in some areas of South Asia and the Middle East. And the majority of that is type 2 diabetes. We've seen a global rise in this over the last few years, and this continues to rise, and it's reaching epidemic proportions. 80% of the cost of treating diabetes lies with complications. So this is things like heart attacks, strokes, blindness, amputation, kidney disease, all of which are entirely preventable with good clinical management. So that's getting clinical specialists to follow the guidelines, screen people appropriately, and start the right preventative medication, and through good patient self-management, through following appropriate lifestyle advice around diet, weight, medication adherence, appointment attendance, and looking after things like feet screening and so on. So the problem is not the knowledge. The knowledge is there to transform this condition and to intervene early and avoid complications. The problem is with the translation of that knowledge into action. So I'm going to give you an example of Scotland, which is the northern bit of the UK, and this is where I've worked for the last 20 years as a clinician and academic. And around 20 years ago, we took quite a different approach to management of diabetes. Like everywhere else in the world, we were starting to see a rapid rise in cases. And we realized that the way we were currently managing the condition was not going to be appropriate for the future. So we took a national approach. We transformed care delivery to make it more equitable across primary, secondary care. We brought in multidisciplinary teams to manage the condition. But the other key thing we did was to set in place a data-driven platform and an information technology platform across the whole country. So now 99.9% of people with diabetes in Scotland sit on our national registry, but not just that, they are included in our national IT system called Sky Diabetes, which is accessible to every clinician across the country who manages the condition. It shares data from the general practitioners, from the hospital clinics, from the laboratories, from the national screening programs, from the pharmacies, to create a single unified electronic health record for the condition that can be accessed by anyone in the country. And it's underpinned by data. And the data within the system, as well as being used for everyday clinical care, is used for national reporting, it's used for world-leading epidemiological research. We've got data on over 500,000 people with diabetes going back 20-25 years, which is fantastic for data mining, artificial intelligence, and predictive analytics, more of which I'll tell you about in a minute. But the other key thing we did was to make all that data accessible to our patients with diabetes. And we do this through a national platform called My Diabetes My Way. Everyone with diabetes in Scotland can access this. It has educational resources, videos, patient stories, e-learning courses. But at the heart of it, patients can also access their electronic health record data. And we make it very clear through the platform, through data-driven targets and visualizations, what the expectation is around achievement of targets like blood pressure, cholesterol, weight, and blood sugar control. In addition, we've been putting more and more data analytics and artificial intelligence into the system so that it will give people automated but highly personalized advice about how they should manage their condition better to reduce the risk of those costly complications. Because we have a clear window of opportunity with diabetes, many years between the development of the condition and the onset of the complications. And that gives us a real opportunity to intervene, improve the risk factors, and reduce the risk of the complications. So over the last 15 years in Scotland, we've seen a huge impact in terms of reduction in the key parameters that are risk factors for complications like blood pressure, blood sugar control, cholesterol level, improvements in key performance indicators. We've seen halving in amputation rates, and the rates of severe diabetes eye disease have dropped to a third of previous rates. In addition, we've got over 60,000 people using the full data functionality of My Diabetes My Way, and we've been able to show significant cost savings because these systems can be run at very low cost because they're highly automated, and we're able to show a 5-to-1 return on investment. So we can save £5 in healthcare costs for every £1 spent on the system. In about 3.5 years ago, in response to a huge demand internationally to have a similar IT system elsewhere, we spun a company out called My Way Digital Health. And since then, we've been deploying our patient and clinician-facing solutions into other environments. So these are now running in around 25% of NHS England. And we're now starting to implement these systems in the US, in the Middle East, and also in India. So we're starting to work on a proof of concept project in Northern India, and I'll touch more on that later. Our patient-facing system, as said, gives access to all educational material for type 1, type 2 gestational diabetes, and for those at risk of diabetes. Everything from e-learning courses to peer support online, care planning documents, and automated advice. Patients can also link up their home data from things like glucometers and Fitbits. And increasingly over time, there's going to be more in the way of Internet of Things data sources that we will be linking into the system. We also collect data from things like patient-reported outcomes. That might be through questionnaires, looking at things like mental health or motivation. And again, that data helps us support people in a very personalised way. On the clinician side of things, the system works as an electronic health record. So great for individual patient management by bringing all the data into one place, but it can also be used for remote patient communication. So we can conduct clinics remotely, we can ask patients to upload blood results remotely, sugar results, and respond to those. And again, during this COVID epidemic, when we're trying to keep people away from clinical services and deliver remote care, that's highly beneficial. The systems can also look at population level tracking. So we don't want to just be able to support individual patient care. We want to be able to look across our entire population. And what that allows us to do through our data algorithms, our artificial intelligence, is to be able to start to pick up who are those patients who are at most high risk of complications like heart attacks, strokes, of coming into hospital over the next 2 to 3 months, of having a severe low sugar event, and so on. And we can pick up those patients early through our predictive data analytics functionality. And that allows us then to recall those patients or to implement certain interventions which we know are going to reduce the risk of those events happening. So this population overview also allows us to track our key performance indicators and quality improvement. So it's a huge driver for quality improvement. If you can't see the data and you don't know where you are in terms of performance, then you can't change it. Whereas if you're tracking it, reviewing it regularly, you can start to implement strategies that really improve the care delivery. The other thing we've done to support mass delivery of education is through something called MOOCs. These are massive online open courses. And we will set up these courses over maybe a 2 or 3 day period and invite people with diabetes free of charge to come and take part in these courses. And by doing this, they will join the course. They'll, similar to what we're doing today, we'll deliver education in a sort of cohort way. There's e-learning resources. There's also live streaming that we do through social media, and they can meet lots of other people online. And we've had around 20,000 people per course interacting with us and engaging with us and completing really very important diabetes education. And this has been a highly efficient way to deliver education across massive populations at scale at a very, very low cost, which in the current environment of a global epidemic of diabetes is really important. So we're, in terms of India, we're planning another one of these open online courses specifically for India in the next couple of months. So follow me on Twitter or Facebook and you'll be able to get more details of that. And that will be something that's culturalized locally and will hopefully support education of tens of thousands of people in a short time. As mentioned, our platforms are starting to roll out internationally with pilots in a number of areas. And when we see the real essence of what we're doing is trying to deliver something at scale that's highly affordable, that can be scaled across entire populations. And as we've done in Scotland, potentially at national level within countries where diabetes is a huge issue. When we talk about our systems, at the heart of it is always the data. So the way we can use data to drive care is really important. And to understand this better, there's something called the informatics maturity model. And this goes from level 0 to level 8. At level 0, we have systems that perhaps generate data. They might be things that monitor blood pressure or blood sugar, but they don't do anything with that data. It sits there unused. Right through to level 8, which is when we're taking information and data from multiple systems. This might be home devices, internet systems, laboratory information, data from the GP clinical record, medication data, and we're joining that data up and we're making sense of that data through algorithms and rules or through computer-based technology. And we're pushing that knowledge back to the frontline staff, back to the clinicians, but also potentially directly back to patients, to allow them to make key decisions in their health. And what we can do is take that down to the level of personalized medicine delivery, where we can start to predict which drugs people are going to respond to and predict which interventions are going to work in different people. We also have something called the 4 Ps of analytics, and this is at the heart of what we do. So when you're using data, it needs to be, think about it being predictive. Can it predict the future? Preemptive, can we use that data to intervene early and stop an event happening? Can we personalize it? Can we use it to tailor our approach to the individual? And we need to think about being participatory. It needs to involve the end users, like the patients and the clinicians, in the application of this. So taking the diabetes context, this is exactly what we've been doing. We've been taking data from home recorded devices, taking data from the medical record around medication, social care, observational data, running this through data analytics. And that's allowed us to generate a system that can help us pick up when we've made the wrong diagnosis, help us decide which of these several medications for diabetes are going to actually work in that particular person sitting in front of you, to allow us to triage people, which during COVID at the moment, we've got to really be focusing on those high risk people because we have limited resource and there are risks to bringing people back to healthcare. But also importantly, we can use data on risk prediction to really help patients understand their condition and drive behaviour change. So we have tools which show patients what their risk of complications is, but more importantly, it tells them how they can modify that risk. What are the changes they can make to their lives around might be weight, smoking, blood pressure, sugar control that will actually improve their outcome. And what this allows us to do is deliver systems that are both scalable and highly cost effective, because there's very little human resource involved in delivery of these systems once they're up and running. And that can mean we can deliver huge cost savings and efficiencies across the service. So I'm going to finish there, and just to say at the heart of, I guess, our journey has been about transforming the lives of people with diabetes at scale through effective, cost-effective, population-based data-driven solutions. And that's always been at the core of what we've done, and keeping to these key principles has allowed us to deliver what we've delivered in the UK and hopefully now transform that into other benefits on a global scale. Many thanks.
Thank you very much. Quick questions, Dr. Wake, let me start with you. So, you talked about 80% of the cost of diabetes being in the managing of the complications. Now, if you were to look backward, how much of, let's say, heart disease or strokes lead to diabetes? I mean, if I were to look backward to forward rather than forward to backward.
Yeah, so I think most of these conditions don't lead to diabetes, but I think what we know is that there's a huge number of people out there who are undiagnosed with diabetes. So a lot of people who are turning up at hospitals with strokes, heart attacks, and so on, often that is the point that diabetes is diagnosed, but they may have had it for several years because until the sugar levels get really high, people often don't get many symptoms associated with it. So often things are picked up late. Sometimes they're not picked up till the point of complications. So as well as implementing systems that can predict things early and intervene, we also need better ways of doing population screening around high-risk patients. And part of what we're doing is trying to say, can we screen populations to pick up those who are high risk and then start to actually screen people earlier so we're not picking them up at the point of heart attacks and strokes.
So would it be correct to say that diabetes is a leading indicator or a key indicator, a pathway to other disease conditions that I will surely have if I have diabetes?
Absolutely. So diabetes is often the initial condition, but alongside diabetes, it often clusters with other conditions like high blood pressure, with heart disease, with kidney disease. All these things cluster together, and often the driver is diabetes. Taking a step back from that, there's also a lot of risk factors like being overweight or not being very active or having a poor lifestyle or poor diet. These things independently can lead to heart disease, stroke, high blood pressure, diabetes. So often the drivers for these conditions are the same. They often come on together. And if we can wind it back, then we can improve not just the diabetes, but all the other conditions that go alongside it.
Right. And these are also the comorbidities which lead to, let's say, COVID patients or being more susceptible COVID patients. So what have you been seeing in the last few months?
Yeah, so it's been really interesting. So we know people at diabetes are at higher risk of poor outcomes with COVID. In certain countries, about 40% of COVID deaths have been in people with diabetes, so disproportionately affected. But it's the patients who are, the risk factors are age, so it's our older diabetes patients, the ones who are more overweight, and the ones with the poorer diabetes control. So it's not just about having diabetes. If you have diabetes but you're controlling it well, and particularly if you can keep your weight within normal parameters, then your risk of complications with infections like COVID is much lower. So it comes down to the same principles around trying to improve it and control it. But age and weight are both significant risk factors. The other thing we found is that your socioeconomic status, so how poor or rich you are if you have diabetes, is a key predictor as well of whether you're going to have good or bad outcomes as a result of COVID. There's less we can do about social status, but it's just another thing that tells us that actually there are big inequalities in terms of social drivers for health as well.
Can you expand that further? So if you were, let's say, more well-to-do, then the chances are higher, or?
Yeah, so if you're poorer, you will have poorer outcomes from conditions like COVID. So we've been tracking our patients during the lockdown, and what we've actually found is those from poorer backgrounds are deteriorating in terms of their diabetes care. Those from more affluent backgrounds are actually doing better. And I think part of this is there's some people during lockdown who have been able to work from home, take the time to actually look after themselves a little bit better, taking the opportunity to maybe engage in more self-care. People from poorer backgrounds often can't afford highly nutritious food. They may have other comorbidities. They're more scared to go outside, and they seem to be reacting in a different way to lockdown, in that they're tending to engage with less beneficial activities. They're putting on more weight, they're eating less well, and they're getting less activity. So we still don't fully understand what that is, but the data tells us that that social status seems to be a big driver towards outcomes.
Right. Dr. Sharma, let me come to you. So I'll come to the second part of what you've talked about and what you've done. And there are lots of questions coming in as well, some of which I will confess I don't understand, but I'm sure you will. So tell us about the rapid detection kit. I mean, that seems to me something that everyone will want to see around them, including while traveling and traveling from one place to the other.
Yeah. So what this rapid test is, so we responded to what was happening in the wider world. We looked at the problems. As I presented, PCR is such a common technique. Every laboratory does it. The problems became apparent because the quantities we work for these reactions, and also the kind of viral strain that we have, because with SARS and the previous MERS, APAS, SOT, there are like 7 strains which are already there in the human population. So which of these is more infective, which is the COVID-19, which is leading to a more drastic response in patients? It's very important to determine what kind of mutations are happening. Whenever the samples are getting positive, they're not always being sequenced because there are multiple steps for reaching that stage of sequencing. So in response to this, we developed this rapid testing because it has got these specific probes, and on the same chip, you can do multiple patient samples. There are 2 probes. So the problem of false negative reduces, and because these are highly complementary to the virus, as it is, the data is being updated, we are creating these probes. So the specificity is very high. It is up to the femtograms, that it's very, very small quantities that we can determine. And we are now working on a 1-step method so that the whole process of heat inactivation, creating the cDNA, could be taken care of, so that in a field, or you can put your reagent, look at the color change, and then determine what's happening. If it is positive, you go ahead and sequence.
Right. So, is this? I mean, is this commercially available? Where are you?
Yes. So we created, we validated it, and we worked with the regulators, MHRA. We got their all advice. So we are bringing it to market. We applied for a government funding because we need to ramp up our manufacturing. So we started approaching the investors as well as the government funding bodies. We are waiting. It has progressed with the UKRI. So we are just waiting their response, and we will ramp up our manufacturing. We want to bring it to the world as soon as possible. Yeah.
Right. Another question, and quick one before I go to the questions that are coming in now. So you talked about drug response and how that was different in different cases. You talked about Asians versus Caucasians and so on. And you also hinted, if I got it right, on the impact of the coronavirus. Would you like to expand on that?
Yeah. So it is again, how the virus is infecting different people. It's variable because there is one of, again, a receptor that is angiotensin converting enzyme, as I was talking about cytochrome. So there is a variability in different ethnic groups. So how these receptors are expressed, there is a genetic and physiological variability. And that is also a determinant. So with, as Deborah talked about, the nutrition, the class, the social impact of it, there are several factors, but one of the factors is also our genetics. So how that interplays, so it needs to be taken into consideration with rest of the studies that we are conducting, and we are studying these patients.
Right. So you're saying genetics does play a role. And I mean, I'm only trying to address the question that many people ask and is very widely discussed, maybe without data, is that, some countries like India, for instance, many people feel we are not as badly hit by coronavirus because of certain fundamental ethnic differences in genetics or genes. So is that on the right track? Is it wild?
Yeah, it is. We have advantages and we have disadvantages as well. As I was talking about earlier, the cytochrome P450 is that in Asian population, we tend to have more toxicities because most of the drug development process takes into consideration the Caucasian patients.
Mm-hmm.
And because over a period of time, there are like 50 genes. So there's huge variability in these cytochromes. So we will tend to have more toxicities. We will have advantages as well. So some of these like CYP2D6, they're well studied, and there are several others that we are just starting to understand now. So
Right.
as we have more data, it makes more sense to understand how this all interplays.
Right. Okay. So let me take some questions. Simonia Vemai asks, with this generation, will cost of medicine reduce in the future?
Definitely it should, because presently there is a long lag gap when we find a drug molecule. There's a lot of testing that's done on cell lines. We are performing a lot of animal tests. If we even reduce 20% of it, the cost does go down. But with more understanding of pharmacogenetics, it's more personalized, more targeted medicines we are talking about. There are new ways of looking at the data. So we are able to look at the interactions, immune response. So obviously, as we make our drugs safer and we understand they can be repurposed, they can be even targeted for certain patients, obviously their cost does go down. Yeah, so in the future, it will be cheaper.
Right. So Dr. Mohammed Abdul Azim asks, how do we make sure the pluripotent stem cell grows into a particular tissue or organ? And once triggered, how do we make sure it stops at the required point of time, it doesn't outgrow?
Yeah. So first is like when we select these lines, we look at the gene expression. So as I was showing the whole complement of the transcriptomics. So similarly, when we make these IPS, we compare them to human embryonic stem cells. So these are highly previously established lines. And then we direct them using small molecules. And at every stage, we again look at the protein expression, we look at the gene expression, and then we look at the morphological features that these cells, they attain. So with these, you're able to direct them towards a particular lineage. And as they attain this lineage, they do reduce the expression of the pluripotent markers. So they will progress from the stage where they can become everything to more of progenitors, and then they become more mature cells, and then they will have attained a functionality.
Right. Okay. Let me switch to diabetes. Parthivi Raturi asks, Dr. Wake, this is for you. How do other factors, and I think you've touched upon this, but maybe it's worth repeating, how do other factors such as high cholesterol and high blood pressure affect a person if she or he already has diabetes?
Yeah. So when we treat diabetes, what we're really looking at is treating reduction in complications. So we don't just treat the sugar level, which is what most people think about with diabetes, but we also look at blood pressure and cholesterol as well, because together, sugar, blood pressure, and cholesterol combined are the three most important risk factors for development of heart disease and stroke and most of the other complications. And they tend to cluster. So as people, if people are carrying too much weight, they develop what's called the metabolic syndrome. And so the consequences of carrying too much weight, particularly around your midriff, so being more of an apple shape and depositing your fat in those areas, will put you at risk both of diabetes, high cholesterol, and high blood pressure. So the three things tend to come on together, and we need to treat the three things together. So if we reduce the sugar level and the blood pressure and the cholesterol together, then we will get a much better reduction in the risk of complications. And so a lot of our diabetes guidelines tell us to focus on those, and we have key targets for sugar control, targets for cholesterol, and targets for blood pressure control. And we want to get them all down to within that target area.
Right, and that's very useful to know. Another question, this is from Neeraj Desai. He asks, is there a global program or platform where governments and companies are sharing data and resources with the idea of finding the best solution to manage diabetes?
Mm-hmm. I'm not sure about a global platform. I mean, I think there are global organizations like the International Diabetes Federation. So they're the, I suppose, the only organization that works at global level around diabetes management. I think there are sort of subgroups within that. There's European diabetes groups, American diabetes groups, and so on. I'm not aware of that. I think what COVID has done is it has brought together some specialist groups globally who are starting to think about these things globally. So I'm certainly linked in with a group that is doing a lot of work in the US and other bits of the world. And this has just been specialist clinicians who have an interest in this area starting to work together. But I'm not aware of a global group around diabetes data where it's all sitting on a platform and we're sharing, but a great idea.
So Manvendra Gokhale asks, interventional medicine requires pharma data touch points and reaction toxicity notations. How does that impact informing patients of the pros and cons with these systems?
So I'm not sure, is that directed at me or Ruchi, but I can say from our point of view, I think what's interesting is in terms of how we deliver drugs to people with diabetes is that traditionally we used to just follow a guideline approach, which would say use drug X, then use drug Y, and then use another drug. There was no personalization to that. So you might end up starting a person on a drug and they get a lot of toxicity from that drug. And actually what we're trying to do now is move to systems where we incorporate data from the healthcare system. So what that tells us is, maybe there was a person who was a very similar genetic type to that person, or their data was very similar, and we could have actually predicted in advance that this person was going to have a poor response to that drug. And I think bringing in the work that Ruchi is doing, what they can deliver is an additional layer above that. We're using the data within the IT systems. If you can also bring in that biological data around the sort of genetics and other enzymes, which would also help predict drug response, then you've got a really powerful system. And I think that's what we're moving towards, is this sort of merging of data and biomarkers sort of into one system that will in the future give us a really fantastic predictability. So we will only be starting people on drugs that, one, they're not going to get toxicity from, and two, that are going to be highly effective for them.
Right. Question from Shama Bingewar. My Health, My Way, is it only for diabetes or other medical fields? And let me complement that with another question. So is there a mother database above the national, is there a national database above this, or does it link to this for health data?
Yeah, so we are starting to work in other areas. So we are doing, as well as diabetes, we're doing diabetes prevention. We're starting to do some work focused on hypertension in people who don't have diabetes, kidney disease in people who don't have diabetes. So we are very much moving into these other disease verticals. And there is huge overlap, obviously, between what we're delivering for diabetes in the system. It can very easily be adapted for other chronic conditions. So we've not yet managed to do that at the same scale for these other conditions that we've done for diabetes, but that is very much the aspiration. And in Scotland, going back to, I suppose, the work in Scotland, we have a pretty good data system nationally that we can pull data for other conditions. It's not quite as good as what we've got for diabetes. And when we're going to other environments, so when we're working in the US, England, India, other places, one of the big jobs that we have is about joining up that data to try to build those data sets. So a huge amount of my team's work is around data integration, linking up data from hospital systems, healthcare systems, devices, and everywhere we go, there's a different data environment. So that's a big bit of work to make the system work. But we're doing that.
Right. Dr. Sharma, let me come back to you. In the beginning, you talked about, it takes 15 years to find a vaccine, and now things are moving much faster. So this is more of a layperson's question. There is a lot of skepticism. There are 135 vaccines now in preclinical trials, 32 in human trials for COVID. And there is a lot of skepticism about various vaccines that have been already announced, because it obviously appears like it's coming too soon. And the skepticism comes from within the medical fraternity as well, apart from, let's say, outside. So all of this creates a lot of concern. So from your point of view as a technology and healthcare person, can you somehow give us more confidence that technology is actually speeding up things so much or so fast that it is possible to get some of these things much faster than ever before?
So in terms of vaccination, some of the vaccine candidates, they have built up on the previous research. So some of these groups been involved researching SARS for many years. So they have not just started all of a sudden. Some of the candidates been repurposed. So how, in terms of immunity, because it does take time for our immune response to come up, because it's the initial response and then the long-term immunity. So how that plays around in short-term studies is to be seen. So there are other technologies, there are methods which are being introduced, as I was telling you about the immune response that are being studied through using the transcriptomics, and there are methods that have involved so we can understand some of these things better. But the pace is definitely faster. And some of these candidates are being introduced for the very first time, like the mRNA, the vaccine strategies and things. So we will have to, I think, wait and watch and respond to the gaps in the technology and build it up to make sure that it's delivered and it's delivered faster.
Right. So Gaurang Kumar Patel asked, Dr. Sharma, the PCR looks great. The PCR lit looks great. I don't know if I got that right. Is sample processing similar to regular PCR that is in practice? Is the probe specific to a virus strain in the UK only, or is it applicable to other strains also?
No. The virus strains, the probes are towards highly conserved regions of the COVID-19. Like the ORF nucleocapsid. So these are the ones which are required by the virus to make its structured proteins. So these are the ones which are also important, like the spike proteins for the vaccination as well. So these are highly specific to COVID-19 as we have looked at the database and the information. So it's not specific to a strain that has been identified in India is different than the strain somewhere. There are mutations that have been identified and more mutations are being reported. So if it is significant, that will be another probe design. But this identifies highly conserved region.
Right. So, there are a few questions, maybe quite a few actually, which are to do with COVID-19 and people, I think, who've either experienced it themselves or family members. Actually, I'm not going to get into that because it might take us into a slightly different area, because we're trying to keep this more technical and academic, and patient advice is something one way or the other, which could be interpreted as patient advice, is something that might be a little dangerous. So let's come back, maybe the last but least question to both of you. And Dr. Wake, let me begin with you. Nitin Naik asks, could you share one example of artificial intelligence-based applications that you would use for drug discovery, clinical diagnosis, or treatment? Any gap you see with respect to how data and information technology falls short versus the hype and potential of such applications?
Yeah, so there are a lot of gaps in artificial intelligence because artificial intelligence is about data. And if we call it, it's the coal that burns this engine. More information we have just seen the results that have come in UK, the bias that was there in the system. Similarly, when the drug discovery data is based on where most of the data or the results or the information is gathered from one gender, obviously the other gender is at a disadvantage. So in the future, artificial intelligence, also it's about automation, how this data is being looked at. So as I was talking about the chip technology, so it's about electronics, so it's the measurements that we are taking, quantifying real-time measurements, the faster processing of cells, because these are very small and they are massive in one field. the tracing of each and every cell. There is huge gap in the technology. So these are being worked on. So it is the new fields of photonics, and so much is happening in the material science. So in the future, but at this stage, I think artificial intelligence, there are no models that you can just adapt, because every cell looks different. So every model need to be trained. So it is about supervised training and then learning through it. So it's a process that will take a few more years.
Right. Okay. Dr. Wake, one example where you've used AI for drug discovery and so on, and what are the gaps that you see?
Yeah. So I'd say not so much in the drug discovery, but where we're using it is really to help us pick the most appropriate drug for applying in that clinical environment. And so we've now got pretty good tools that will tell us which of the diabetes drugs we've got are going to respond in certain individuals. But I agree with Ruchi. The issue is it's the data that you've got. So the data, you know, and we take it into different environments, we might or might not have all the data applicable. I think what will move this on a level is when the genetic data becomes more accessible. So at the moment, we very rarely do genetic testing on people with diabetes. But as genetic testing for drug response becomes more widely available and more cheaply available, and we can start to pull genetic data in on top of the biochemical data and all the other data we've got, that will make a huge difference to these. So that will take time, and it's really about price point, I think, to bring these technologies down and them starting to become more applicable generally. And I think some of the other barriers are around the regulations. So we're deploying a lot of AI at the moment, but for us to be able to safely put that into clinical practice, we need to step through a lot of hurdles in terms of testing, clinical safety testing, and then get through the regulatory authorities, whether that's the FDA or the MHRA. And that's all done for good reason, but it can mean that actually there's a big time from something being a system being produced to actually reaching the market at scale, because of all these regulatory boundaries and barriers that need to go through. So I think speeding up of that process would really help as well, bring some really innovative stuff to market a bit quicker.
Right. And there was a question earlier, which I think you referred to, but is there a timeline for what you're doing in India? And you said that there is a program or initiative?
Yep, so we've got some funding from Innovate UK to do some pilot work in India. So we're working with a couple of key providers, big healthcare organizations in India, to do some scoping work to see whether will our systems actually work in India? Is there the data maturity of a level in the healthcare system at a level that our systems will actually work? We'll complete that project in December, and then that will move into a second phase where we'll start to actually implement some of our systems and evaluate them initially within a couple of key pilot centers. And then if that works, we'll expand from there. So we're working with partners at the moment to do that, but we are going to be putting out an open, free, massive online education course for people with diabetes in India in the next couple of months. So as I say, if people want to follow me on LinkedIn or Twitter or whatever, I will give you more information as that comes, and it would be great if people can share that. As I say, it's completely free, and we're just trying to really get to the good information out there to help people.
Right. That's a wonderful note to end on. There's a lot of information available on the work that Dr. Deborah Wake is doing on diabetes, on managing diabetes. And of course, if you want to find out more about rapid testing, you want to find out about what's happening on cutting edge research in stem cells and so on, you should follow Dr. Ruchi Sharma. We are talking about health and technology, and usually in the past we've talked about health, we've talked about epidemiology, and all oftentimes in the context of COVID. So the future of health, which is what this is about, is obviously very encouraging, and it's always great to speak to people who are at the cutting edge and who are showing the light, so to speak. And I do wish both of you, your teams, and your colleagues all the very best in finding solutions and fighting not just the various conditions that you are fighting, but also obviously COVID-19. On that note, thank you very much. And that's it from me. And it's back to my hosts at TEDx Gateway.
Thank you for having this. Thank you for joining us today, Govind, Ruchi, and Deborah. It was an amazing conversation. We're still getting in questions, but I'm sure the attendees out here can reach out to you on Twitter or Facebook or LinkedIn, or if you could share a place where they can write to you, because we're still getting many people asking us questions right now. There are quite a few unanswered ones, though. I'm really sorry about that. But again, it was an amazing session. And I look forward to hosting each one of you again for the future of health in the future, because what we're doing right now is we're hosting multiple experts from different parts of the world in this series. Every fortnight you will see session, you will see speakers coming across and talking to people from the industry. So do join us as attendees. We'd love to see you on the other side. And thank you for taking out time today. Thank you again, Govind. Thank you again, Ruchi and Deborah.
Thank you. Pleasure. Take care.